All right, we are recording.
Okay, so we did our presentation on Enterococcus faecium and facialis, and I'm Summer, and with me is Susie and Alyssa.
Okay. So for the morphology, Enterococcus faecium and facialis are both very similar. When it comes to their morphology, they're both gram-positive. They have a coccus, or in other words, a round shape, which that can be seen in Diplococcus or Streptococcus arrangements. They are both also non-modal. There are not many differences between the two pathogens, but it can be seen that Enterococcus faecium can live in either aerobic or anaerobic environments, while E. facialis is facultative anaerobe, meaning that it is not necessarily restricted to that environment. The top picture is an example of a slide from the faecium, and the bottom is an example of facialis.
And for their metabolism, they are both pretty alike as well. They both go through the glycerol catabolism, where glycerol is their main carbon and energy source, using glycerol catabolizing enzymes through the intracellular growth, and they both do follow anaerobic and aerobic conditions, as said before, using ATP-dependent glycerol kinases, and they don't have the Krebs cycle involved, so they do lactic acid fermentation, and as said before, they do the facultative anaerobe.
Okay, so for the diseases, Enterococcus faecium is generally found in intensive care units. It's resistant to active substance classes, which can also be known as it being resistant to substances like penicillin and other drugs. It causes diseases like neonatal meningitis, which is when the meninges, or in other words, the protective membrane in the central nervous system, become inflamed, and this can cause mortality. This pathogen can also cause endocarditis, which is also another form of inflammation, but this time it's referring to the heart's inner lining, or in other words, the endocardium. The Enterococcus facialis is known to cause contamination problems in hospital and institutional settings. Urinary tract infections are the most common infection. They are non-seasonal, which means that they're usually hospital acquired because of the antibiotic resistance of the pathogen. Another problem is that it is so resistant to antibiotics. They're usually ineffective, which is what allows it to have the ability to actually cause diseases. And then lastly, some diseases that this pathogen causes are endocarditis, like I said, sepsis, meningitis, UTIs, and other bodily infections.
So known antibiotic resistance for Enterococcus faecium, this bacteria is a multi-drug resistant, just like facialis. One thing that the faecium can do is create a biofilm to help resistant. They're resistant to ampicillin and vancomycin, and more resistant to penicillin than Enterococcus facialis. Facialis is also resistant to quinupristin, dalfopristin, and vancomycin. And it can be treated, it has some susceptibility to ampicillin and penicillin, but it's really rare, and can also be treated with piperacillin. Just a few things for both of them. The resistance in a few of these strains usually have mutations in the protein sequence, and at spot 485, instead of a met, it's alanine, and a serine substitution in spot 466 in the loop structure or the penicillin binding protein complex. And since I said penicillin, it is mostly for the microbes who are very high penicillin resistant. Another thing that confirms the resistance that we can tell whether the microbe is either resistant or not is if beta-lactamase is present. This just helps us to see that it's actually really rare. So what are we using in the lab that's the safer relative? It's the Enterococcus raffinosis. They're also gram-positive, and it is circles, so it's cocci, and it usually goes into chains. And these are the sites that we used. All right, thank you.